From Microbe TV, this is a special immune booster recorded during the American Association of Immunologists conference in Chicago, May 2024, starring Cindy Leifer and Brianne Barker, and featuring Heather Caslin from the University of Houston.
Welcome.
Thank you so much. Thanks for having me.
Yeah, great. Usually we like to start off by asking our guest a little bit about how they got started in science, how they get sort of interested as far back as you'd like to go.
It's funny because looking back now, I can see it earlier than I think I did at the time. When I started figuring out what I wanted to do in college, I think I didn't know right away, but I ran track and I danced. So I was like a ballerina all growing up. And so those are what I really love to do. And my senior year, I got injured. And so I ended up in physical therapy and had to go through the recovery phase. And I think as I was thinking about what I might want to do as a career, I was like, oh, it might be nice to help other people kind of get through an injury. So I actually went to school interested in physical therapy. I went into exercise science as my major, which was really fun, learning about physiology through kind of the perturbation of exercise itself. And then I actually had applied to physical therapy school and done the interviews and gotten in. And kind of right towards the end, I was invited to a research conference. And I didn't really know what research was before that. A little bit was going on in the department, but it wasn't super visible. And the couple times I had kind of tried to see what it was about, it just kind of didn't work out well. And I didn't pursue it too hard. So as I started to I went to this conference and I really started to think this is really, really, really interesting. This isn't what I thought research was at all. I need to take a pause for a second. So looking back, it was kind of that intro from exercise into physiology and then kind of into looking at research.
So did you turn down, did you turn down going to physical therapy school? And did you just work in a lab? How did you transition then?
I did. So I feel like I had a 1 week midlife crisis because that that deadline was right there. And I was like, wait, I need to learn about this other thing first. And then after talking to a lot of people about what life is like as a grad student and what research is like and all the different types of research, I kind of came to the conclusion that if I did a master's, so I ended up being able to do a master's in the same department with the same people who kind of knew my story and knew what I needed.
And where was this?
Virginia Commonwealth University.
Okay, cool.
And so they kind of knew what I needed out of it and what I wanted to do. And I said, worst case, if I hate it, I go back to physical therapy and now I have a master's, so they'll probably like me still. But best case, maybe I like this. And I think about 2 weeks into a summer research project that I started helping with, I was like, nope, this is it. I'm not looking back. So
awesome. So what types of things were you working on there?
So that project itself was looking at kind of compound stresses. So the stress of waking and adiposity, how that impacts a second stressor. So we did exercise and we did psychological stress testing and we did a couple different stressors. And so that was really the thing that started. It was really working with blood and then starting to do ELISAs on it and things like that. That I was like, ooh, this is what I really like.
So you were measuring immunological markers as well as stress markers in the blood with different stresses and exercise and with people with different weight gain. Can you just talk a little bit more? Because that's not something that we usually think a lot about.
Yeah. So we were mainly looking at cortisol and some of the stress hormones. We did norepinephrine and epinephrine ELISAs. And I think they had it's probably IL-6 and TNF were two of the immunological markers that they were going to look at because those are kind of the first, right? So we started looking at those. And as I started doing a thesis work, I started adding on, I think I added IL-1 beta and then substance P, which we didn't end up measuring. But I started kind of looking in what else could I look at? So it's funny you say that those aren't things we normally look at. I took a immunology course in the course of the master's project to be what are these cytokines? I don't know what they are. And after the immunology course, I was like, I still don't know what they're doing in any of these disease states, right? Because in the immune course, you kind of have to start with the cells as they respond to bacteria and viruses and parasites and maybe cancer now. But that's kind of where the first immunology course ends. So I said, I still don't know what's happening at baseline or to mental stress or to chronic disease or to none of those are covered. So that's what really fed me into okay, you got to do a PhD if you really kind of want to know the answers to this.
So after you did the master's, did you stay or did you go somewhere else for your PhD?
So I actually ended up staying at the same university, but changing departments.
Okay.
And I worked with a mast cell immunologist, and that was just kind of by chance, I think. So now mast cells are one of my big loves. But basically, in coming into it, I knew I didn't have a lot of the preparation that I think others might have had going into a umbrella type program at a medical school for immunology.
Okay.
Because I came from exercise science and didn't have that as like the end goal right away. So I knew this professor. I knew that he was a really, really good mentor. I knew that his students felt supported and happy, and I kind of knew a couple of them.
Mm-hmm.
And I knew that like he knew where I was. I was really proud of the like 6 ELISAs I had done. And then I'm like, oh, your lab does that every day. Like everyone's doing that regularly. But he kind of knew that that's where I was. And I think looking back, maybe I could have had a little bit more like self-confidence and like other people might want me to.
So what types of things were you interested in terms of the immunology when you were in your graduate program?
So I was really interested in the idea of exercise immunology. But at the time, everyone kind of doing that as like a whole field. They were coming from the physiology world. So all of them had been kind of classically trained in exercise science and exercise physiology, and they were learning kind of immunology as they went, which is one great like side of the project. But I figured if I got trained really like more hardcore in the immunology side, I would have just something different to contribute to that field. And so I figured that just it ended up being mast cell biology, but looking at the cell itself and looking at signaling pathways and learning the techniques would be really important for that journey.
So did you look at it first in exercise, or did you look at it in other model systems?
So I actually haven't gone back to exercise yet.
Okay.
But I just got a project that will start like this summer. So finally, like we're coming back.
Okay.
But in my PhD, we did, the whole lab really was looking at how mast cells respond to the environment. So we did things like TGF treatment, and I ended up doing lactic acid treatment or lactate treatment for my PhD. So looking at how in asthma, in sepsis, in allergy, and a lot of these things, also in cancer, lactate's really high in the environment. What is that doing to mast cell function? And kind of another arm of our graduate lab was looking at how like repurposing drugs could affect mast cell function. So we were looking at statins and SSRIs is kind of where they moved after I left the lab.
So can you say a little bit more about that? So repurposing drugs, they have direct effects on mast cells, and you were trying to suppress them or activate them? Like, because
so we thought about it a lot in the context of allergy. So more trying to suppress the mast cells. Mm-hmm. Being in the US, that's more the issue that we see.
Please suppress my
people
allergies.
Around us. Well, the first AAI conference I went to, actually, I was presenting a poster, and the only comment someone had was like, mast cells, those are awful. Those give me allergies. And like, why do we have them? Which we have them for parasite purposes. There's still, and probably a lot of things we don't know. I think they're really understudied as far as like tissue homeostasis, since they are in all the tissues. But yeah, the suppressing allergy is kind of the main thing mast cell biologists study at this point. But statin drugs they traditionally for lowering cholesterol, but they actually have a major anti-inflammatory effect as well. And so likely in patients who take them, there is a cholesterol effect, but there's also this effect on the immune system that's really interesting. And kind of people are starting to gather that side of things.
Right. What about lactate? What types of things did you see with the mast cells there? Because I could kind of imagine how that might relate to exercise.
Yeah, exactly. So when I kind of came in, I was like, ooh, I know what that one molecule is and like how to get it from like lipolysis. So I was like, I feel like I can like jump into that. But the lab had kind of seen that lactate is anti-inflammatory, which others have seen in the context of macrophages and other cell types. And so we did find in mast cells too that it's anti-inflammatory to IgE activation, so allergy type activation. And then I was doing LPS activation. So sepsis patients actually have higher lactate levels. It's likely in response to their kind of overactive immune system that happens early on with the cytokine storm, but that likely is contributing to the immunosuppression that they see as that cytokine storm goes down. So we did see that that's anti-inflammatory on mast cells as well. And so in exercise, I actually think that like with high intensity exercise, you have an inflammatory response that's productive for muscle kind of healing and muscle building. But the lactate may kind of work to suppress that. So I haven't done that piece of it with like, if you take lactate away, what happens to that inflammatory response? But I do think it plays a role there too.
Awesome.
That would be cool.
So what kinds of models were you using to look at the mast cell response?
Mm-hmm. So a lot of it, what I liked about the lab is we kind of had a general way papers were kind of done. It was a great training way to like jump into immunology, where we did a lot of like the first figure, we did a lot of like dose response time course, look at all the different cytokines that are produced. look at the mRNA and the like ELISA data. So you can look at protein and mRNA. And then you kind of work towards like, what is signaling doing? And then at the end, we always ended it with a mouse model and some human data. So with mouse models, you can do passive systemic anaphylaxis is the big kind of IgE-driven allergy model. We did some like IL-33 induced peritonitis when we were looking at IL-33 activation of mast cells. And then I actually did an LPS sepsis model. So not quite as relevant maybe as bacteria itself, but it did give us that inflammatory response and some of like the cytokine storm driving piece of the sepsis.
Awesome. So what happened after your PhD?
So in studying the lactic acid stuff, because I'd come from the exercise world, I'm like, it has to be metabolism. Like it's suppressing metabolism. So I got to do a tiny bit of that. We got to do like a seahorse experiment early in some of those studies, but I really got interested in the idea of immunometabolism. And so one arm of that is how immune cells metabolize glucose and amino acids and fat, right? The other side of it is immune cells actually have this big impact on systemic metabolism. So I actually applied to labs or kind of sent out feelers to labs that were on both sides of that coin. And I ended up really falling in love with a project with Alyssa Hasty at Vanderbilt University. So she was looking at weight cycling. So a lot of people look at weight gain and the impacts on immune cells and kind of diabetes development and other cardiovascular diseases. But her lab was doing like models of basically yo-yo dieting, which are really common. And like, it sounded really relevant to me with like the people I knew and just the environment that we're living in. Every diet that comes out, people jump on it and come back from it. So that project was kind of personally interesting and also just like professionally interesting, I guess. But I got really interested in the idea that like cycles of weight gain and loss have a different impact on your immune system than just adiposity itself.
Okay. What types of impacts are those?
So what we see, some human studies support this, but when our mice do yo-yo dieting, so we do low fat and high fat diet. So it's all like diet driven itself right now. We see that they have worsened glucose tolerance or diabetes risk compared to mice that have just kind of steadily gained weight over the same amount of time. And humans actually see that as well. There's some higher risk for like hypertension, some specific cardiovascular things. I think it's really understudied actually because it's hard to do. It's hard to follow patients for 20 years or go back into medical records and really get an accurate sense of what that weight looks like over time. But what's really interesting is we find that after weight loss, systemic markers of metabolism all improve, which matches kind of what humans see and kind of why weight loss is prescribed to the general population for different people. But adipose immune cells stay more inflammatory. So they never really come back to their lean state. They stay more inflammatory and kind of, it's more of a Th1 environment after adipose expansion and weight gain. But they never kind of go back to that more Th2 regulatory cell type. So they kind of remember that state of higher inflammation. And we've actually seen there's a CD8 T cell memory response there. So we think there is some adaptive actual memory developing. And then my project became innate immune memory. And we see some of that too. So there's like actual immune memory. And then there's kind of like the idea of this memory.
So they remain more inflammatory, like as a marker, or they are continuously producing inflammatory marker, like cytokines and things?
So at least on the innate side, so I've been
potential?
looking at macrophages. With macrophages, the potential after weight loss is higher.
So
if
then
you
when
give them a second stimulus, that's
so
when
if
you really
regain,
see it.
then they're more inflammatory.
Then you do see a baseline more TNF production.
Okay, so that's why the yo-yo is a problem, because when they're high in
after weight loss,
they,
there's no like if there's no signal on them, they're just kind of primed and ready.
Right.
But when there's another stimulus like weight regain, or we've done some like bacterial activation, they're actually more inflammatory.
Does that also sort of increase the propensity to gain with the weight back?
Probably, but I would say we haven't done like a study that would actually show that. So potentially, I do think like relevance to the world wise, I think it helped explain why it's so hard to keep it off.
Absolutely.
So I think for patients, I hope that's, I mean, it's kind of nerve wracking that that's true, but I hope it explains to people that like there's these real biological processes that make this really hard. And it could be targetable. I mean, that's the big thing, right? If we could target some of these development of memory in this context, I think that could be really applicable for people.
So probably not related necessarily to what you were studying, but one of the big things it's everywhere in the news and so many people are doing it. And I'm sure you know what I'm going to ask. It's like all these weight loss drugs that are causing rapid weight loss, but they're changing the physiology really with GLP-1s and things like this. So has anybody started to look at what that might have, impact that might have?
I think a year ago or so, the sense was like they're not impacting the immune system. They're definitely just on the brain, the hypothalamus, and the pancreas, and some of these. And I'm not sure that's totally true. I think that's still to be
surprised. I mean, immunology underlies everything, right?
Like a little bit of PubMed searching on that, and there's to it.
I think by gene expression, you kind of don't see the classical receptors all the time, but we know that that's not the only way you can do stuff. So I think the effects on the immune system, we still don't know yet and need to be studied. It
would be interesting, though, to look at your memory and your innate memory that you're seeing and see if it impacts the development of that, because it potentially has a lot of, has a potential for yo-yo, right?
Yes.
But as soon as people go off, they may regain.
We do know that for most people, unless there's other interventions that people start dieting more or doing different diets, different exercise, different drugs after that if you just come off the drug itself, you regain, which is scary because they're really expensive. So if you change insurance, if you have side effects from them, there are people who probably will, especially those who are like going on it more for like personal reasons than necessarily medical reasons. So yeah, I think that it'll be a really important thing to study coming in the future.
And sort of the flip side of that is we often think about the sedentary people, but what about athletes? Is there anything different about their physiology and immunology? Because that's sort of how you got into this, because you were so active and interested in the physiology.
I am interested. So one of the studies we just got funded for are to put exercise into our model. So if you're doing exercise during either weight loss or weight regain or all along, how does that change? Because exercise has specific effects on like exhaustion in T cells as well as like macrophage activation. And so I think exercise could, I'm like 50-50. Every time I read the grant, I was like, is it going to be better or the same? Hopefully better. But so theoretically, if exercise is impacting these pathways, maybe it wouldn't have as dramatic an effect on somebody. But I do get the question, like in sport athletes who like make weight classes, like wrestlers do a lot of like quick weight change. I mean, I think the thing we don't know in humans is like the degree of it or how much weight has to change, or if it matters if it's muscle or fat or what other type, if it's all water weight, like those things we just have no idea. So I think the application is still, I can't say much about. Right. Talk to your doctor, right? But but there's a lot of questions kind of that way that would be interesting to study.
So after your postdoc, what happened next?
So we kind of wrapped up those studies kind of nicely, and it kind of fit in timing to like start applying for faculty jobs. So I started my position in September at the University of Houston. I had applied to both like immunology departments because I feel like that's where I feel like I'm an immunologist now, but I also kind of wanted to go back to the exercise science world. So I do want to ask more questions related to exercise over the course of my career. But I kind of liked the idea of like the people in that field doing exercise immunology are all physiologists. So I still feel like there's some kind of unique perspective I can bring to it that'll be fun. And I do like thinking about teaching. I'm excited to teach undergrads is really like the population I wanted to go back to. And I think there's a lot, especially with like, so you guys are science communicators, you know a lot of this, but like in how to approach science, how to talk about it, and how to teach students to talk about it, and how to teach students to like interpret what they're seeing on TikTok or Twitter, right? Like some of that, like I can actually, I feel like in the realm of health and fitness and immunology, like you can find relevant articles every 2 minutes, relevant like things. So
awesome.
So can you say a little bit more about what it was like to start your lab? Because we haven't talked to that many people about who are this new. So you're about like 8 months in.
Yeah. It's been, I'll just say first, it's fun. I think you've been working so long for this. You're kind of like, okay, this is it. Like now I get to try it. It always feels slow. At least like I haven't talked to anyone who said it feels like they got started fast. Yeah. Because you're like, I bought this fridge. Now it's going to take a couple months to get it. And I bought the centrifuge. Now I have to wait. I think you see, like I knew about animal care protocols and like health and safety protocols, those type of things, but without doing it yourself and really like needing to wait on your research to start till they're there. I think the waiting has been a little bit new. I think you learn a lot about, I think every university, it seems, functions a little bit differently in like pre and post award. Like there's a lot that you learn right away. And then I think the emails, I like knew PIs had to do a lot of emails. But again, until you're the one like doing it, it's a little different. So it's been learning, but. And
you said you already have one grant that you're going to be working on, some of the things that you described?
Yeah, I got a couple pilot grants in the fall that'll help do an exercise study. And then I really want to look at the intrinsic metabolism of the cells because a lot of that is what's driving innate immune memory. The epigenetic remodeling that occurs is driven by some of the metabolic pathways. So we'll do some metabolomics and lipidomics as well.
Have you started hiring people for the lab?
I have 2 part-time technicians. So they're in kind of like the post-bac phase. They both want to go to medical school. And one is like taking a class and one is kind of working else on the side. So what was hard right away is I was like, okay, I found these 2 great people, but like, wait, now I have to wait for my or these grants. I'm like, I technically got one in December, but we still don't see the money yet. So I'm like, oh, okay, let's go. And then like, okay, wait, we have to pause. So I'm actually kind of happy that they were part-time right away because it's hard to give someone 40 hours of work, right? When your lab is still really new. I feel like the hardest part is you can't offboard much or you can't like delegate much right away when you don't even know what you're doing. So that part's been hard, but I'll have a graduate student start in the fall.
That's exciting.
Which is exciting. So by then I think we'll be like fully up and running. I mean, we have some cells, we have some mice, so things are starting to happen.
Awesome.
It's not a surprise. It's always slow at the beginning. I remember that. I think I relied a lot on my colleagues to be able to let me like
use
run
their centrifuge or something.
Yeah, right. Use their equipment and as long as I had my incubator and a hood and a little bit of reagents, I kind of bummed everything else at the beginning to keep experiments going.
Well, then I feel like you have to I'm like, okay, let's just like replicate everything else first. Like we can't even do something new until you do the old stuff.
And it's surprising sometimes depending on what you do, if it doesn't exactly repeat, like with mouse experiments with variations in facilities can sometimes set you back a little bit. So hopefully all of that goes well.
Good luck.
Are you going to do any human stuff as well, or is it almost all in mouse?
Yeah, so one of the kind of big draws that I liked about University of Houston, it's part of the Texas Medical Center. So Baylor College of Medicine is down there and MD Anderson and UT Health and a whole bunch of them. So there's a bunch of people at Baylor who I've been talking to who we could either get samples from the blood after weight loss, or bariatric surgeons are the person who can, I think, get adipose for us the best. So hopefully we'll start to like set up those collaborations more formally. But yeah, I think the application to humans is the most important piece of this because if it's all mice, then what does it mean, right? So we'll see.
That's awesome. All right. So this is a very random question.
Yeah.
On some of our other podcasts, we will ask people what you would have done if you hadn't become a scientist.
I think there's like, I actually find things all the time that I'm like, ooh, that's really interesting. We were talking about this yesterday with another new person who's transitioning from postdoc to PI. Like, it's really scary. What happens if it doesn't work out? I'm like, I have a 3-year-old, so we go to the zoo all the time. And I'm like, this would be fun. I could come like work with animals every day. Like that's been the most recent thing that I'm like, okay, if this doesn't work, I think the scary part of being a PI is once you have people who rely on you for their salary and their degrees, like that piece would make that hard. I couldn't just like leave tomorrow and not feel bad about it. Right. But there's like so much interesting stuff out there. And even science communication, during the pandemic, I got really interested in it. And it's really hard to do while you also have another job. So I commend you guys a lot. It's amazing. But yeah, I think there's...
So not a ballerina.
Not anymore. Though, funny enough, the first time I came to Chicago, since that's where we are now, was to audition for the Rockettes.
Really?
I did that at the end of high school, and I was in there for like 5 minutes, and they were like, no. But that was fun. So actually, it's still a positive memory of Chicago that I have. So.
Awesome. That's exciting.
That's really neat.
That was a like short-term dream, but
so fun. Is there anything else that you would like to tell us before we wrap up?
I guess in thinking about, I was excited to meet you guys today because, I mean, the idea of science communication to the general public is really big. But as someone, I've actually never formally been in an immunology department. I was in like health and human performance, and I've been in a physiology department for my postdoc, and we were technically in like biology for my PhD. So I've had like access to seminars and immunology folk the whole time. But I think even like not having necessarily immunology colleagues all the time, being able to like listen to the podcast. I'm like, ooh, I love hearing how it's relevant to teaching, right? Like, hey, this is where I'm going to put it in my course this year. And oh, this applies to that other paper we just read last week. Like hearing some of that on a regular basis is really fun. And it's a really great resource to send to students, as well as your YouTube videos. So
I'm glad they're helpful.
Those are great.
We were actually talking after an earlier interview about ways that I'm trying to figure out how I'm going to be using some of these interviews in my classes.
Oh, yeah.
Yeah. Can you say more? You said you're going to try and incorporate.
Yeah, I haven't fully fleshed out this idea yet, but I'm starting to think about pairing some of our interviews with relevant lectures, maybe having the students read a paper from the scientist that's related, and then listen to our interviews since they're kind of short. And think about the topic that way, but also get to see a human behind the things that we're learning in class. So I haven't fleshed the idea out totally yet, but it's something I'm thinking about.
Yeah. I think it's a great idea that we can use what we're doing here even to benefit what we do outside of this and teaching and so forth. But it's great that you're a listener and that you're excited, and we were so glad to have you on today.
Great. Yeah, this has been fun.
Yeah, this is awesome.
It's nice
Great.
to meet you guys in person.
That's a special episode of Immune and Immune Booster. If you have questions or comments, please send them to immune@microbe.tv. If you enjoy our work, please consider supporting the science shows of Microbe TV. Go to microbe.tv/contribute. Microbe TV is a 501c3, so your donations are U.S. federal tax deductible. Our guest today is from the University of Houston, Heather Caslin. Cindy Leifer is at Cornell University, at Cindy Leifer on X. Brianne Barker is at Drew University, BioProf Barker on Blue Sky. Music on Immune Boosters is by Tatami. Thanks for listening to Immune, the podcast that's infectious.